Hello and good morning, everyone. Welcome to the fourth and final episode of Tuesday's series with New England Biolabs NEB, brought to you by NextGen Scientific in partnership with NEB. Today's event is about Build Your Next Generation Sequencing Library with NEBNext. My name is Fateen and I will be serving as the moderator for today's event. Before we begin, I would like to seek for everyone's cooperation to put your mic on mute. I would like to also remind everyone that today's series is interactive and we encourage you to participate by submitting as many questions you want at any time you want during the presentation. I believe our speaker would be happy to answer them all after her presentation. Today we have invited a speaker from NEB Singapore to give the presentation on NEBNext products, which is Ms. Geun Ju. Geun Ju, if it's possible for you to share your your camera later on, so our attendees today could see your face.
Sure, I'll do it right now.
Thank you, Geun Ju. Right, so Geun Ju started working at NEB Singapore in July 2019 as a field account manager. So she mainly supports customers from local research institutes, biotech and molecular diagnostics companies for the entire NEB product portfolio. So in addition to that, she has been conducting training sessions and giving presentations specifically on next generation sequencing and amplification related products. So prior to joining the NEB team, Geun Ju worked as a project manager with a sequencing service provider, Macrogene APAC, and has done an internship with Illumina Singapore. As for her academic background, she graduated from the National University of Singapore with a Bachelor of Science with Honours in Life Science. So without further ado, I would like to welcome Ms. Geun Ju, who will take us to explore the workflow of next generation sequencing with NEB products. Let me hand it over to you, Ms. Geun Ju.
Thank you, Fateen, for an awesome introduction. Yeah, let me try to share my screen. Just give me a minute. Okay, are you able to see my screen?
Yes, very clear. Thank you.
Right. That's great. All right, so I'll start my presentation. Good morning, everyone. Thanks, Fateen, for the introduction. I really have nothing to add on about myself, but instead, I'd like to take a minute to introduce our Singapore team. Just hold on for a while. Yeah, so with me today, we have Dr. Go Song and Dr. Jonathan Hao. Song is a key account manager looking after Singapore account with me. She has a very extensive research experience as she did her PhD in NUS and worked as a research fellow in the same lab prior to joining the team. And she used our NGS products for her thesis and projects, so she has a very good hands-on experience. And Jonathan, he's our technical applications manager, who is the superhero behind answering all the technical questions coming in from APAC countries, including Malaysia. So the team is here with me today to address any questions and queries that you have regarding our products or NGS in general. So do feel free to leave your questions in the chat box, and we'll be happy to answer all the questions at the end. Yeah, all right. So with this, I'll start my presentation, NGS and products from NEBNext. The subtitle NEBNext is the combined word for NEB and Next, which stands for the next generation sequencing. And this is the product name that we use for NGS kits and modules. This is the overview for today, and I hope it will help you to navigate through the presentation. So first, I'll give a brief introduction of NEB, and second and third will be the topic of our interest today, NGS, and its workflow. And point number 4, as part of the NGS workflow, I'll elaborate on the sample prep step and introduce our product, Monarch. And subsequently for point 5, I'll break down the details of library prep steps and also share the relevant products from us, branded as NEBNext. And for those who have been doing NGS extensively, may find this part of presentation pretty basic, as this contains an introductory information. But please stay with me. There are many points and products that will be unique and interesting to you. And number 6, I will show you how different applications like DNA sequencing, RNA sequencing, methylation sequencing, ChIP-seq evolves around one central workflow using our NEBNext Ultra core reagent. And then point number 7, I will share some of the products that are used for molecular diagnostics, including NEBNext products. And lastly, I have a slide for COVID-19 research highlights. Yes. NEB is founded by Don Com in 1974. This is the picture of him. And since then, we have placed science and environment as our highest priorities. And we became known as an organization by scientists, for scientists. And NEB's philosophy can be distilled down into these 3 core values: passion, humility, and being genuine. So just the 3 points out of many that defines us. We have first scientific expertise with over 45 years of experience in this field, having more than 1,300 publications to date. And second, we are known for an excellent product quality, service, and value. So we are ISO 901 and 13485 certified, and we also have GMP facility up and running. We are very committed to the environment, sustainability, and also giving back to the community. So our good citizenship has been recognized, and we became a certified B Corp early this year. And it is a certification that's given to organizations with highest standards for social and environmental performance, transparency, accountability, and it goes beyond the product service, and it takes into account of employees, customers, community, and environment. And this is the timeline of the innovation that took place in NEB. And it's my last slide for the introduction. So we released the first commercially available recombinant protein in 1980s. Yeah, and something relevant to our topic today is that NEB launched NGS sample prep and library prep portfolio in 2009. So we've been supporting NGS field for more than 10 years. Next, let's move on to next generation sequencing introduction. So what is, just hold on, yeah, okay. So what is NGS? NGS is a massively parallel sequencing technology used to determine the order of nucleotides in entire genome or targeted regions of DNA or RNA. And it offers ultra high throughput, scalability, and speed. What does it mean by massively parallel and ultra high throughput, right? So before NGS was widely adopted, capillary electrophoresis-based Sanger sequencing was used. And it was used for the well-known Human Genome Project, which took over 10 years and costed nearly about $3 billion. However, NGS enables scientists to analyze the entire human genome in a single sequencing experiment or sequence thousands and tens of genomes in 1 year. So the words massively parallel and ultra high throughput here really well describes the characteristic of the robustness of NGS. And it has a various applications, and I've gotten this from Illumina website. And certainly the areas of application is not limited to this, and it is evolving. And but I want to just highlight these things here. So it allows us to rapidly sequence whole genomes, deeply sequence target regions, for example, whole exome sequencing, and allows us to discover novel RNA variants and splice sites. Also quantify mRNAs for gene expression analysis. And it allows us to analyze epigenetic vectors like genome-wide DNA methylation and DNA protein interactions. And I'd like to highlight that whole genome bisulfite sequencing has been the gold standard for this, but we do also have a kit called EM-seq, utilizing enzymatic methylation, preventing the DNA damages. You can actually find more information about this, and if those who are interested, do let me know. I'll share more details about the EM-seq. And NGS also allows us to sequence cancer samples to study rare somatic variants, tumor subclones, and etc. And study of human microbiome is getting more and more popular, and metagenomic and metatranscriptomic works are ongoing. And with the COVID-19 pandemic, NGS became a critical tool to identify novel pathogens and detect its variants. This is the history of NGS platforms. So the first generation is the classic Sanger sequencing using chain termination method. And then it moves on to the second generation, which we call the next generation sequencing. And it is predominantly represented by Illumina sequencers. So Solexa was the pioneer of the NGS platform provider, and it was later acquired by Illumina. And Illumina sequencers leverage the sequencing by synthesis, SVS technology, which allows to track the addition of nucleotides as the DNA chain is copied. And this is being done in a massively parallel fashion. And third generation platforms are long-lived sequencing platforms like PacBio and Oxford Nanopore. These came in 2010, and they can read about 10K base pair DNA fragments or even longer. And this can improve the sequencing coverage in the regions of the genome that were previously inaccessible or difficult to analyze with the short read sequencers from the second generation sequencing. So for the genomes that are not known or without the reference genome, hybrid of second and third generation is often used to have a complete map. And the focus of today's presentation is about the sample prep and library prep for the second generation Illumina sequencer, which is still considered as the gold standard. This graph shows how the sequencing cost and data output changed since 2000. And there you can see that there has been a dramatic rise of data output, which is on the right side of the y-axis, and then drop in the cost of sequencing, which is on the left side of the y-axis. Applied Bioscience introduced the first automated capillary electrophoresis-based sequencing instrument, which you see, it's actually AB370 in 1987, and then they came up with ABI 3730XL in 1998, around 2000. And these were the primary workhorses for human genome projects that I mentioned just now. And while these first generation sequencers were considered a high throughput for their time, Genome Analyzer by Solexa emerged in 2005. And this massively parallel sequencing technology revolutionized sequencing capabilities. And from this time point onwards, the data output of NGS has been more than doubling each year. So with this now, let's take a look at the NGS general workflow. So when I put it very simple, it distills down to the 4 steps: sample prep, library prep, sequencing, and data analysis. NEB provides solution for the first 2 steps, sample prep and library prep. And these are supported by our 2 product portfolios, Monarch and NEBNext. And this entire workflow will vary according to your choice of the sequencing instrument. So now I'm going to show what options we have for the sample prep. First, from the crude sample, we need to extract gDNA, total RNA, or high molecular DNA to construct libraries before sequencing. So first, we have Monarch Genomic DNA Purification Kit, which can purify high-quality gDNAs from various sample types. And second, we have Monarch Total RNA Mini Prep Kit that extracts total RNA from different samples like cells, blood, saliva swabs, tissue, bacteria, plant, and many more. And we have the new addition to this portfolio. It is a high molecular weight DNA extraction kit. And this utilizes a novel method using a glass bead to extract ultra high molecular weight DNA, which is useful for long read sequencing like Oxford Nanopore sequencing. And with this, let's look into the details of library construction steps. And for today, I'll focus on the library prep for Illumina sequencers. Once we have the input material, such as extracted gDNA or total RNA, we can construct the libraries with it. And this involves a coordinated series of molecular biology reactions, which are fragmentation, N repair, detailing, adaptive ligation, and PCR enrichment. And these are done using the reagents that NEB has been supplying and researching for 45 years. And these are such as polymerases, ligase, and polynucleotide kinase. So let's look at the first step, fragmentation. What is fragmentation? Fragmentation is the separation or breaking of DNA strands into pieces. And there are typical methods of fragmentation, which are nucleation, sonication, which is mechanical shearing, and enzyme-based method. Mechanical shearing using Covaris is the gold standard for fragmentation. It makes it easy to control the fragment size. However, there are some disadvantages, such as upfront instrument cost, consumable cost that's reoccurring for tubes and plates. And it is indeed time consuming, especially when processing large batch of samples. So it's difficult to scale up. And reaction volume is limited, and it causes a DNA damage. So what NEB has as an alternative is the Ultra II fragmentation step. It utilizes the enzyme-based fragmentation combined with end repair and detailing step in single reaction tube. So it's capable of generating a wide range of insert sizes by varying the incubation time. It's robust and easy to use and compatible with standard DNA buffers. And it gives high yield and results in high quality libraries. There are some disadvantages, like concerns that cleavage is not being very random, or transposes having very specific input requirements. So the choice of the fragmentation method will depend on the user's priority and preferences. This graph shows that our fragmentation system ensures a consistent fragmentation size, regardless of the input amount, ranging from 100 picogram to 200 nanogram. Next step to the fragmentation is the N repair step. So what is N repair? It generates a blunt-ended fragment by filling in the 5' overhangs and chewing back the 3' overhangs. And this is done by T4 DNA polymerase with 5' to 3' polymerase activity and 3' to 5' exonuclease activity. And 5' phosphorylation is done by T4 PNK. Next is detailing. It's a creation of single A 3' overhang, and this is critical for the next step, adaptive ligation, where the adapters have the single T overhangs, which is complementary to A. As I've mentioned, adaptive ligation is the ligation of adapters onto the A-tailed fragments. And you may ask, what is the adapter? And it is a short sequence-specific oligos that are complementary to the sequence present on the PCR primers, which is very critical for the next step. This slide compares the Illumina's adapter with ours. And as you can see from the diagram, Illumina has the Y-shaped adapter, whereas ours have the hairpin loop structure, as you can see. And this shape increases ligation efficiency and minimizes the adapter dimer formation. And to make this fragment ligated with this adapter accessible for PCR, we need to remove the uracil in the NEBNext adapter by user enzyme. And we are ready for PCR enrichment step. So what is PCR enrichment? It is the amplification of adapter ligated DNA fragments. And this process incorporates barcodes. Barcodes is actually the same thing as indices, and which is the same as index. And this is to enable multiplexing, meaning pulling the samples, different samples together. And it also incorporates P5 and P7 sequences that are required downstream. I will elaborate this in detail with this schematic diagram. So first, you see the adapter ligated fragments in the middle. And there is I7' on the right N on the left. And if you take a look at the detail, index primer contains the barcode sequence, which is denoted as BC here. And this is the same as index sequence. And it also has the P7 sequence, which helps to fix the library fragments into the flow cells, because this has the complementary sequence to the oligo sequences on the surface of the Illumina flow cells. And next, we incorporate the universal I5 primer, which are also complementary to the oligo sequences on the Illumina flow cells. And as a result, after this PCR cycles, every individual samples will have its own defined barcode or index that will allow you to pull or combine the samples, and we call this as multiplexing. This slide depicts the various multiplexing options that we have. So first, on the top left, we have single index primers with an index length of 6 base pair. There are set 1 to 4, and this allows us to pull up to 48 samples. And another single index primer we have has the index length of 8 base pair, and this allows you to pull up to 96 samples. This comes in a 96 well plate, where each of the wells contain combined primer mix of universal primer I5 and the index primer I7. And at the bottom, we have dual index primer options. So primer set 1 to 2 is available for the 8 base pair length primers. So in this case, you will receive 8 vials of 5 I5 primers and 12 vials of I7 primers. And this can be combined to make up to 96 reactions, 96 combinations. However, if you need to pull samples more than 96, you can use both set 1 and 2 to combine everything and pull up to 384 samples. And finally, the unique dual index primer pair set has index length of 8 base pair. And these contain 96 unique combination of I5 and I7 primers. It's available as a sealed 96 well plate. And we recommend you to use this if you're using a sequencer with patterned flow cells, for example, NovaSeq. All right, these are the illustrations of the multiplex oligo index options in 96 well plate on the left and in vials on the right side. And I prepared following few slides to highlight how different library prep kits evolve around one central workflow, NEBNext Ultra II core reagents. We call the core reagents as the heart of the matter because different applications like whole genome sequencing, chip sequencing, exome sequencing, RNA sequencing, and other workflows that we have are built on top of this Ultra II core reagents. And the characteristic of this core reagents are, first, it saves the time. It's very user-friendly protocol with modular workflows, and it allows automation with various liquid handlers listed here. And second, it reduced the cost by making it simple and allowing us to have minimum pipetting steps. And most importantly, it allows customers to generate high-quality libraries with minimum input and give a maximum yield. All right, this slide shows the NEBNext Ultra II DNA library prep workflow, which essentially use the Ultra II core reagents. So each green arrows will be very familiar to you. It's something that we visited earlier. And what's special about NGS, NEB's NGS product is that we made this kit available as the modules as well. So the customers have option to buy the entire kit or the modules, individual modules for each steps. I'd also like to highlight that we have kit options with the purification beads. It's especially useful for smaller labs where they don't have bulk purchase of the cleanup beads from Beckman Coulter. So on the top left, you can see again the Ultra II DNA library prep workflow with the individual modules indicated as a green arrow. And I'm going to show you that this workflow is incorporated in each of the applications that are listed here. Yeah, so now on the right-hand side, it shows the workflow of different library prep kits, which are fsDNA library prep, enzymatic methyl-seq workflow, RNA sequencing workflow, and low-input RNA library prep workflow. And I hope you already recognize that they all have something in common, the green arrows. And the green arrows are the part of Ultra II DNA workflow. And as you can see, for example, let's say for RNA library prep workflow, it's about 50% of the workflow that overlaps with the Ultra II DNA workflow. So it's going to be extremely useful if you're working on multiple applications, because once you're familiar with this one Ultra II core reagent, which means the Ultra II DNA workflow, you became an expert of other workflows as well. And it makes it very easy for you to pick up on other applications. And I believe this is especially relevant for anybody who works in a sequencing core, where you have to handle various applications. For the next slide, I will share some of our products that are used for molecular diagnostics, including NEBNext products. So due to COVID-19 pandemic, sequencing of SARS-CoV-2 virus has become more important. So our amplification and NGS reagents are being widely used for virus detection and sequencing. And these are just to list a few. And we also collaborated with RT Network to produce RT kits, which are compatible with Illumina and ONT Oxford Nanopore platforms. And these are based on the original work of the RT Network. And the kits include balanced RT primer pools, RT-qPCR reagents, and library prep reagents. Lastly, I'll share some COVID-19 research highlights. Please note that this is very much of an evolving area and information is constantly changing. But for now, there are several applications of sequencing for SARS-CoV-2 studies. First, epidemiology and surveillance study is the major part of the application. But also basic research and clinical research is also being done. And for diagnostics, amplification technology like PCR and LAMP is still the more popular and the gold standard method. But now antibody and antigen detection test is also widely adopted for virus detection because of its speed. So less usage of sequencing for the diagnostics for now, for the ones that requires very fast turnaround time. But definitely it is having more and more critical roles as well. So it's very exciting to see how sequencing can be applied. And customers who are involved in COVID-19 researches are individual labs, core labs, sequencing service providers, and COVID-19 consortia. So there are a huge amount of international collaborations that's going on. And there are a number of methods for SARS-CoV-2 virus studies, and the main ones are listed here. So first, targeted virus sequencing is common for sequencing the actual virus. And when people are looking more broadly than the actual virus, they are looking at host genomic and transcriptomic sequencing, as well as immune response related sequencing. And it's just to add an interesting work that's done by the researchers from Stanford Uni. They used leftover droplets in vials of Moderna's vaccine to determine the sequence of mRNA for SARS-CoV-2 spike protein that's used in the vaccination. And they explained that knowing the vaccine sequence will allow diagnostic labs to more easily differentiate between RNA from the vaccine versus from the actual viral infection. So I find it very interesting and wanted to share as an additional point. And there are several high-profile preprints and protocols that uses our reagents. So from Sanger Institutes, Cornell Medical College, Decode Genetics, And we have collaborated with many other industry partners like Oxford Nanopore to come up with the kits. This is from our website. So products that support COVID-19 research is available in this one landing page. And it's an evolving list, and updates are being done time to time from our marketing team. And new protocols and preprints are available from the web page. And just to show you here, I've just got a screenshot of the papers and preprints that utilize our reagent. And you can see that the second one with the SARS-CoV-2 enrichment sequencing by spike primer, MSSPE method version 4, it utilized our Ultra II RNA library prep kit for Illumina sequencing, as well as the multiplex oligo that we have talked about. Materials are abundant, so brochures for each applications like DNA library prep, RNA enrichment method, and enzymatic methylation sequencing and so on is available. So please request to our distributor, Fateen, and yeah, she'd be happy to provide. And we are happy to support them as well. And tech notes are available, which illustrates the performance in detail, especially with comparison to the kits from Illumina and Roche. And the details of the automation compatibility and the app note is also available from our website. Link is below. So for example, Biomax, Hamilton Star, EP Motion, Tech and Freedom Evo. So these are some of the instruments that we have validated, and we can check with the team from these companies on the compatibility if you have some automation plans going on. Yeah, and that's it for my presentation, and thank you for your attention, and I'll open up the floor for the questions.
Hi, Geun Ju. So thank you for the very informative sharing just now. So we've got a question from Ria Ilani, Miss Ria. So we are planning to use NEBNext companion kit for ONT. What is the quality requirement for the RNA input, as in read and purity?
All right. So quality requirement for the RNA input, read and purity, is that right? For the ONT?
Correct. Yeah.
Yeah. So exact number I am not able to recall, but this is available from our website. Let me just let me just have a quick search on this. And I can share the product page as well. Okay. This is our product page. I might have to check the Oxford Nanopore page for the exact protocol. So yes, maybe can I get back to you again with this?
I see. Because I think researchers have this I would say challenge when they're dealing with clinical samples, especially COVID. Yeah, so I think to get the high quality read and purity would be a challenge. So if you have any tips or tricks for that.
Right. So yes, this module actually contains the reagents for FFDNA repair mix and FFP DNA repair buffer. So definitely it does address some of the concerns that you have. And yeah, I actually checked the website FAQ section, and the protocol is available from ONT website. So let me just give you the this is the number that you can search from ONT website, on how you can use our reagents,
Right. Okay.
and using their technology.
Thank you so much.
No worries.
Yeah. So do we have any more questions from the floor? You can, just type in the chat box, or you can turn on your microphone. Geun Ju would be very happy to answer the questions.
Yes.
Yeah.
Maybe if anyone can share their experience using our NEBNext kits and how they have been successful, it would be also very helpful and encouraging for us.
So Geun Ju, thus far, do you receive any how do you say, questions from Singapore sites on using the NEBNext Arctic?
So far from Singapore side,
Yeah.
not yet, because I believe the sequencing work is really more actively done in US and Europe. So yeah, haven't heard of like any customers using RT kits for SARS-CoV-2 sequencing in Singapore. What about in Malaysia? Is there anyone who's using this kit?
We do have, and then actually Malaysia is going to start with the 3,000 genomes very soon. So I think we do need the support, especially from if customers are using NEBNext companion kit Arctic. I mean, they're just about to start, so now it's like I think the actual questions would come in when they started using the kit, and I mean, like maybe then we should seek for your tech support.
Right, that's right. So we have Dr. Jonathan Howe. He will be happy to address the questions, and it's actually being more actively done in US. So we have product managers and product supports who can better address the questions for the RT kits. So we have different line of support. So Jonathan will be the one who is receiving the questions. And we are not able to resolve the issue. We have the product development team who actually came up with the products to really address the questions to the customer. So yeah, good line of support that we have here.
Yeah, that's good to know. Awesome. So we've got a question from Sean. Say, could you explain how RNA from COVID and RNA from mRNA vaccines are differentiated using NGS?
Oh yes. This was just one of the article that I briefly read and wanted to share about it. So I think it's to determine the sequence of mRNA for the SARS-CoV-2 spike protein that's being used in the vaccine, and they will actually see where this comes from, whether it's from vaccine sequences or from the RNA from actual virus. But yeah, it is a good question. Thanks for the question. Let me dig on that paper again and find the details. If anyone has any suggestions or ideas, please feel free to speak.
Right, I believe that would be the final question, Geun Ju and everyone. Thank you again, Ms. Geun Ju, for answering the question.
Oh, sorry, Fateen, I have another question from Sean.
Yeah, I just saw, sorry. Yeah. So, a bit about adaptive ligation was too fast. Could you go through that again, Geun Ju?
No problem. Thanks for the question, Sean. So let me share the screen again. Right. Are you able to see the screen? So, Sean, if this is the part that you have the question on, basically, you see that the fragmented DNA fragments are here, and we do DA tailing, and this is to, sorry, it's not proceeding. Okay, this is to ligate the adapters with the T overhang, so it will be complementary to the A. So the whole fragment will be DNA fragments with the adapter ligated together. And this adapter is crucial, important, because it contains a sequence that's complementary to the PCR primers that's required on the next step. So let me see. So you see that these are the adapter. If you see can you see my pointer? I'm not sure if you're able to see my pointer. Okay, once again, so these are the loop adapter that we actually cleaved using the user enzyme, so it became open, and it's complementary to the index primers. So this will be incorporated through the PCR cycles, and these index primers are important because it has the barcode sequence, which is the same as index sequence. It's like a name tag for the libraries. And then it also has the P7 and P5 sequences. It's complementary to the oligo sequences that's on the Illumina flow cell. So basically, the libraries can be fixed and be held on to the flow cell using this P5 and P7 site. Yeah. So Sean, I'm not sure if this answers your question and it clarifies your thought. Do let me know if you need any further clarifications. All right, thank you.
Right. So I'm not sure if we have any more questions. Do we have any more questions?
Hi, I'm Hasana. Can you hear me?
Yes, hi Hasana.
I'm going to ask regarding the companion kit.
Companion kit for Oxford Nanopore?
Yes.
Sure.
And yeah, it's for Arctic SARS-CoV. Regarding the protocols, you mentioned that there are two different protocols for the. The first one is express, and the standard one, which is either with the cleanup or without the cleanup. Yeah, and then may I know that maybe from your previous clients or any reports, how does these two protocol affects the sequencing results, or maybe one has better, or you maybe have reports from here?
Sure. So Hasana, if I can clarify your questions. So you're referring to the Arctic SARS-CoV-2 library prep kit, is that right?
Yes.
All right. So is it for Illumina sequencing or Oxford Nanopore sequencing?
It's for Oxford Nanopore.
Okay, sure. Yeah. And your question is that there are two types of protocols, express and the standard,
yes,
and how this usage of these two protocol results are like, is that right?
Yeah, because like in your protocol, it's just mentioned if you use express, you just like reduce your hands-on time, and then and something related to the related to the technicals. I mean, like improve your coverage and everything. But at the end of the sequencing, maybe does it affects the sequencing result?
This part I'm not very sure about it. Is it okay if I get back to Fateen and Fateen get back to you?
Sure, sure.
Yeah.
Hi, Ju, can I add on?
Yes, sure, Jonathan.
So what you say is correct. As written in our protocol, when you do express, it's faster, and your sequencing coverage might take a bit longer to get sufficient coverage. And so this is actually the main answer behind it. And the main reason why we actually have an express protocol is because, as you know, our Arctic COVID-19 test kits are actually based on the Arctic network protocols, and they actually had developed Express kit, and many people were actually asking us whether or not our kits can use Express. And we actually collaborated with the scientist Josh Quick from University of Birmingham, who is also one of the main scientists behind this Arctic network. And so we did develop an Express protocol with the main intention to just remove the cleanup steps. But as to the main difference is actually you do need to sequence a bit longer to actually get more coverage for each sample. As to the quality, I don't think I can comment on that until you try out. I could not find any questions or answers or comments from my team based on my searching through my database.
Okay, thank you.
Yes.
Thank you so much. Another thing, so that is there any like preferred method that from your clients either use Express or standard?
So okay, right now this Arctic protocol, especially this kit, is relatively new, but I do have clients from other countries which are also using it. So far, most of them are still using the standard, because the Express, they will want to try the standard first before they go to the Express. It's just a difference in cleanup step at the moment.
Or any from your technical team, do you guys try both of them, or?
So all the protocols which are published in the NEB, so for this Arctic companion kit, we actually did have testing out, and it works, but we do not recommend one over the other. It really depends on your purpose, on your preference, because especially when you use the standard protocol, you actually do get better coverage, okay, in a shorter runtime. But there's another group of customers who actually wants a faster timing, because if you do the Arctic COVID-2 standard protocol, it will probably take like 5 to 6 hours just on the preparation. So some customers will want to do it faster, and that's where you get the Express.
Okay, sure. But none like reporting anything like the sequencing result is better using one or another?
I do not have any information on that. I will try to check it out, but so far I do not have any information on that.
Okay, sure. Thank you.
Mm-hmm.
Right. Thank you, Ms. Hassana. Thank you, Jonathan and Geun Ju, for answering that. But we will get back to you, Ms. Hassana, on the answers once we get them.
Sure, Fateen. Thank you.
Thank you. So I think that would be the final question. Yeah. All right. So thank you again, Ms. Geun Ju and Jonathan, for answering the questions. It was an absolute pleasure to have everyone with us today. From the very first series, PCR, then qPCR, to CRISPR, and today NEBNext. In case if you would like to revisit this webcast, we have recorded this video and we'll make this available on our YouTube channel in a few days to come after this presentation, and we will notify you. Wow. So we welcome all of your feedbacks by filling in the feedback form, where the link can be found in your chat box now. So, and then due to high interest of COVID-19 related topic in the previous series, we are currently scheduling with Jonathan to give the presentation on NEB supporting COVID-19 research. We will blast the information soon as everything is finalized, so keep your eyes peeled for our update. So thank you everyone, till next time. We hope you have a great day ahead. Thank you very much.
Thank you, Fateen. Thank you, everyone.
Thank you, Geun Ju.
Yep. Bye-bye.
Bye-bye.